This section will introduce vision. If you look through the syllabus of the course, you'll notice that vision takes up about half the course. Vision is a big area in sensation and perception. It's probably the most studied. Vision is probably the most developed in the primates. It's a very complex perceptual system that we have, and you probably take it completely for granted. You never sit there and think about how great your vision is, but it's really remarkable. Vision also takes up about a third of your brain, the back part of it. And it includes things not just, sort of just seeing the world. It includes things like reconstructing perceptions of the world in 3 dimensions. It involves integrating vision with other sensations, especially somatosensory sensation, or your touch sensations, so that you can orient your body in 3-dimensional space. It involves a lot of memories about things that you see, a lot of memories for visual experiences. You have, of course, names for things that you see. You integrate a lot of information with the visual environment, and all that's done by a large part of your brain. Well, if you look at this picture here, you see the brain with its covering still intact, and that way the eyeballs could still be in place. And there you see the brain with its coverings. You see some of the veins and arteries that cover the dura. That's the membrane that you see there. But look at where the eyeballs are positioned. I mean, the eyeballs are extending out from your brain. Your eyeballs are not separate from your brain. It's all integrated into one visual system. It's as if your brain has this special receptor for light information, and it incorporates it intimately with brain function itself. And I think when we, look about the world, or we see people with vision problems, we tend to locate vision in our eyes, but that's just a small part of the visual perceptual system, and the brain is often not thought of as the processor for visual information. Okay, well, we're going to cover this, just the basics of vision, just the basics of some of the anatomy of vision, and focus this part on also more on the eye than on the visual pathways and perceptual systems that are in the brain. We'll cover those as the course goes along. Right now we're just going to deal with the receptor system that's in the eye. The first thing to understand about vision is to understand the nature of light. Light is an energy that radiates from a light source, and it's part of the general electromagnetic spectrum. It's a part of the electrical field that's generated by any electrical activity, but of course light is in a special part of that spectrum that we can perceive, and we call it light. But actually there's no difference between light, what we call light, and FM transmissions, or television transmissions, or your AM radio, or infrared transmissions. They're all part of the electromagnetic spectrum of frequencies. The only difference between them is the frequency range that they participate in. So X-rays are at certain frequencies, gamma rays are at certain frequencies, infrared radiation is a certain frequency, radar is a certain frequency, and what we call light is a certain frequency. Light just happens to be the frequency that we can see. Now imagine if you could see radar radiation or FM radiation. So if you go to an airport and you see the radar device moving around there, and that sends out a transmission of radar waves that bounce off objects and come back to the detectors. Well, imagine if you could see that. Now, presumably, if you could see it like you see what you call light, you would see it as a color of some kind. Maybe it would just be white. It could be a frequency that your brain colors. At any rate, you'll see it like you see light. You see it like you see sunlight or light coming from a lamp. You would see it radiating out as if the radar device was a big searchlight. Imagine if you could see FM transmissions from radio stations. I mean, all of you use cell phones, you all use radios. All those represent electromagnetic radiation in a certain part of the spectrum that you can't see, but your electronic devices obviously can detect it. Your radio can turn that back into voice or whatever was sent by the FM transmission. But imagine if you could see it. Imagine if you could view FM transmissions. So you would look around, you could look around the city, and you would see where there are radio stations. You'd see bright light there. You'd see it attenuated as it goes across the landscape, as if it was a big searchlight. It was a big, big light out there. Well, all that is just to sort of make the point that the radiation that we see is just a part of the spectrum, this same spectrum that's used for all these other purposes. And we've been able through history to detect these radiation frequency levels and be able to use them for various things. So we use them for radar, we use them for FM transmissions, for radio transmissions, we use them for cell phones. You can imagine how we've been able to modulate these transmissions and be able to use this information, all starting around the turn of the century. We really had very little idea about FM and TV transmission ranges until Marconi and some of the others figured out how to use radio. Okay, we see the spectrum here at the bottom. You see the colors that are in the spectrum. Now, it's important to keep in mind that the colors are not in the light. The colors are in your brain. And what you're doing, what your brain is doing for you, I don't think we all have a conscious idea that we're doing this. What your brain is doing for you is mapping out certain frequency ranges as certain colors. And by doing that, your brain gives you a schematic, sort of a drawing of the world that's colored by the frequencies of lights that are out there. Now, it's a very powerful tool to have this kind of mapping of frequency. But you should begin to think of it as a perceptual phenomenon and not something that's actually in the world. So your brain is mapping frequencies as colors. And here we see the spectrum of colors, the rainbow of colors. Now, a lot of research has gone into examining what colors are and so on. The earliest research that was done by Isaac Newton. And he sat in his little room in Cambridge and darkened the room and made a little light source in the window, a little pinhole in the window, so a little beam of light came through. And then he began to study light using prisms. And what a prism does is create a medium that separates the frequencies. And so when you see a prism, it looks like it's creating a rainbow. It's actually separating the frequencies that are in white light. And Newton was the first to figure that out, that white light represents the mixing of all the colors. Now, that doesn't seem to make any sense. When I think about white light, I don't think about it being a blending of colors. Because whenever I've actually done that with paints or something, it comes out a very mottled brown color. The way you should think of white light is think of it as noise. And if you imagine noise and hearing, well, imagine it's the blending of all the frequencies of light. And you see that noise, you perceive it, your brain maps it as whiteness. At any rate, colors are mapped by frequency, and we'll get into this in much more detail later. It maps out a certain narrow band of frequencies in the electromagnetic spectrum. Now, an important concept for understanding the spectrum is that light is transmitted as waves. And it has a wavelength. And here you see the depiction of that in the slide. A wavelength is a way of measuring the frequency. So if you've got a frequency there, say in the green range of 500, that means that the wavelength is 500 nanometers. So the distance from one hill to the next is 500 nanometers, which is a very tiny unit of measurement. Now, as you go up to the red range, you see you got 600 nanometers, and so the wavelength is now longer. It's now broader. And as you go down, you see that you've got more narrower toward the blue range of 400 or so. That's 400 nanometers, and that's even smaller. Let's take visual perception a little bit farther and present another general concept, or a set of them, really. Here you see a slide that indicates the world. I refer to it as the world as it is. So here you see a picture of a butterfly. Now what reflects off the butterfly is just frequencies of light. There are no colors in the butterfly. When your brain perceives these frequency differences, it's going to map them as colors. As you go to the next slide, you'll see that this happened, this mapping of frequencies as colors. In this slide, we see the mapping of frequencies as colors. So light frequency differences have now been perceived by the visual system. Your brain then schematically changes the world as it is into a world that now has colors in it, representing these frequencies. Now, the invention of colors is something completely done by evolution over the course of millions of years. It obviously enables you to discriminate different kinds of objects from each other with much more accuracy than if you only saw them in black and white. Here, for example, you now can distinguish this butterfly from some other kind of butterfly that has a different color pattern. Now, through the evolution of butterflies have evolved to have patterns of substances on the scales that make up their wings and so on to reflect different color patterns, so they can be recognized by each other, so they can warn predatory insects or predatory animals to stay away from them, and things like that. So color plays a very important part in the visual environment of every organism on the planet, and humans are very, very good at color perception and color mapping because our visual systems are so sophisticated. Now go on to the next slide, and you'll see another artificial manipulation of visual perception that's done by your brain. You may have noticed when you went from the previous slide to this one that the image of the butterfly became sharper. And that's another thing that your brain does, your visual system. It will sharpen images that are naturally blurry. The world is actually blurrier than you perceive it. So the light, when it's being reflected off objects, is actually a little noisier than your perception. And largely through a process of lateral inhibition and other processes in your brain, visual information is processed in a way that emphasizes contours of objects. And so images are sharpened so that you see a figure sharply against its background. And obviously that helps you adapt to those environmental conditions where there are contrasts, where you can see sharply where an object ends, for example, if you're going to pick it up, or if you're going to do something with it. You need to know where the object begins and where it ends. In that way you can manipulate it better when you orient your body to it. So sharpening is a very powerful tool for making objects in the world appear clearer than they actually naturally appear. Here are some more elemental aspects of light that have a lot to do with how light deals with either reflection off objects or media that it passes through. And these are just four aspects. The first is refraction, which you've all probably experienced if you go into a swimming pool. You'll notice that the objects below the water appear differently than the objects do when they're above the water. And what happens is the water medium bends the light, and it bends it in a way differently than how it's been traveling in the air. And so this is called refraction. So as light goes through different media, it will refract. It will change direction based upon the medium that it's in. And refraction is, in a way, a kind of reflection, really. It's just that the medium is changing. It's not that there's an immovable object in front of the light. Another thing that happens is reflection. Now, reflection is an important aspect of all activity of light. As light hits objects, part of the light is absorbed and part of it's reflected. And in the bottom left, you see a depiction of absorption. As light hits an object, the object's properties, its chemical makeup, will cause some of the frequencies to be absorbed by the object, and the remaining frequencies will be reflected. Now that determines the frequency that's reflected off objects that your brain perceives and maps as colors. And so if I see something like a red apple, what's happening is the light frequencies other than red are being absorbed by the skin of the apple, and the red frequency has been reflected by it. As if you had a mirror there that it only reflected red light, and all the others are absorbed. Well, my brain, of course, your visual system is only going to see what's reflected off the object, and that frequency is perceived by your system and turned into redness. And so your brain maps out that part in which those frequencies are present as red. The final property here is diffraction, and this is a property where light is dispersed when it goes through a small aperture. And that's not so important I don't think, for human perception as the others, but it is an important property that makes light become blurry and more difficult to perceive. Now we've gone through some basic aspects of what light is. Here we have the receptor for light, the eye. Now we all know how important the eye is in perceiving light. It has a certain basic layout, and I'll go into more detail as we go along. The camera model works pretty well to describe this, since people are mostly familiar with cameras. You all know that a camera has a lens on the front of it, and this lens is going to focus light onto, in the old days it was focused onto film, now it focuses onto receptors in electronic devices that perceive the light level and frequency. But the focusing aspect is still there for the lens. You still have to focus the lens, focus the light using a lens on a certain surface that's going to perceive the light frequency and the light level. And here you have it with the eye. So you see an object out here, the lens focuses it back on the fovea, which is the central part of vision. It's the acute area that higher mammals have for perceiving light very accurately. Notice that the lens, however, turns the cup upside down. Now, all your vision is actually upside down. Now, I don't perceive my vision to be upside down. Your brain sets it right back up, because obviously you've grown up with this apparatus, and your brain knows that the world is actually right side up and not upside down, produced by the lens. I think I'm going to show you a movie later that depicts people wearing special prisms that actually turn the world back around again, so everything looks upside down initially, but they adjust to it very quickly, and they begin to live in the world as if nothing's wrong. And I think it's because the brain is already doing the flip-flopping, and it just needs an orientation point. And once it gets used to that, it makes everything depicted correctly. At any rate, you have the basic system where you've got light impinging on the lens. The lens focuses it back on the retina, and the receptors cells in the retina are going to be perceiving the light. This is a blow-up of the retina. Now, the retina is a special layer in the back of the eye that has all of these light receptors in it. And here's the basic layout. Let's go from the bottom and go toward the top. At the bottom layer there in blue, you see the receptors. Notice that there are rods and cones, and I'm sure you've heard of this before. But there you see a distribution of mostly rods and a few cones in there. And the nuclei of these cells is above the receptors. And so that layer that comes next is called the outer nuclear layer, but it's where the nuclei of the cells are. The next layer up is referred to as the outer plexiform layer, and that's where you have cells that interact with the receptors. You can see one there interacting between these receptors. And these cells actually facilitate some of the initial filtering that goes into perception to sharpen images and things like that. Then they're connected to an inner nuclear layer of these intermediary cells, and you see that what they're going to be doing is basically notice that there's more receptors than there are these other cells, indicating there's going to be some kind of convergence of the receptors onto these cells. Then there's another plexiform layer where you have an interaction between this layer of cells. You see that green cell there showing there's an interaction between them. Then finally you have the ganglion layer. The ganglion cells are the nerve cells of vision. The ganglion cells are neurons that connect to this system and finally get input from it. then they convey information all the way back into the brain through the optic nerve. Their axons are going to go all the way from these cell bodies you see here, all the way into the middle part of the brain, the lateral geniculate body, and then that's going to go back into the brain to the occipital lobe. And we'll get to that in more detail later. Right now, what you want to see here, what you want to notice here, is that there's a receptor level, there's an initial processing level, and there's finally a level where that initial processing is conveyed to the central nervous system. And the other thing you should notice about this is that light passes through these upper levels before it gets to the receptors. That, unfortunately, is an artifact of how the retina had to develop in an evolutionary sense, and just how it had to be structured. But unfortunately what happens is, of course, these upper levels will diffuse the light. And so some of the blurring that happens and misperception occurs because light is displaced and diffracted by having to go through these layers up top here to finally get to the receptors. But at any rate, this is the layers of the retina. You can see it's relatively simple, even though it's not just receptors and it's connected to neurons. There's some initial processing that goes on in the retina, mostly to sharpen the image. And then finally this sharpened image is conveyed to the central nervous system, where it's further sharpened and then processed for form and color and all kinds of things. But at this point, the nervous system is just conveying frequency data from the cones and luminance data from the cones, and then just luminance data from the rods. And we'll talk about that a bit more in subsequent slides. Here you see the layout of the retina in a schematic. And so you have photoreceptors, you have horizontal cells and bipolar cells, amacrine cells, then finally you have the ganglion cells at the ganglion level. So the horizontal cells, the bipolar cells, the amacrine cells, they perform an initial processing of the visual information so that it has high integrity by the time it gets to the ganglion cells. The ganglion cells are then depolarized. The little conical shapes that you see at the end there represent the terminal parts, different cells, as they are synapses, and they depolarize each other to finally produce a depolarization of the ganglion cells. When that occurs, the pattern that's fired up into the brain represents the frequency of light and its luminance level, its intensity level, being conveyed up into the nervous system for processing. Okay, here are the receptors you've been hearing so much about, the rods and cones. And I'll be going into much more detail about these when I go into the color section. But this is just an introduction. Now, with rods and cones, what you have is basically a specialized nervous system cell that's going to be specialized for responding to different light frequencies and to different light intensities. Now, the rod is the more simpler of the two, and the rod is going to respond to just light intensity level. So you can think of it as black and whiteness, grayscale level. A cone has special pigments in it that responds to a different light frequency. Now, each cone is sensitive maximally to a certain light frequency range, which I'll get to in a second, but they're tailored for light frequency discrimination and only respond to a certain light frequency range. Then both connect to the ganglion cells, to the horizontal cells and so on, through the same system of synaptic terminals, just like a neuron. And so when one of these fires, it's an indication there's a luminance in that part of the retina. So when the lens projects light back on the retina and one of these fires, it suggests a certain luminance is there. The rod is just going to be responding to the luminance level, the intensity level. The cone is going to be responding to the intensity level for light within a certain band of frequency. And I'll get to that in subsequent slides. Here's a depiction of the fovea. Now, the fovea is an indentation in the retina that represents the point at which the lens is supposed to focus the light. So this is like the back of a camera, in which the lens has focused light onto the surface of film. And here you see the fovea as this indentation. The fovea structure allows for a maximum density of cones. And only cones are in the fovea. Now, cones are the more sophisticated receptors that allow for the perception of light frequency and luminance, and they represent our very acute, sharpened vision. There are some people, I have a movie about this, there are some people who are born without cones, and so their foveas are essentially blank there, and they have great trouble with reading and doing anything that requires very fine vision because they don't have the acute vision that comes about with cones. And of course, they don't have any color vision. They perceive the world in black and white and grays. At any rate, though, the fovea represents this special spot on the retina that represents the most acute vision. It's where the lens focuses the light and where we have only cones. Now, there are lower mammals that don't have foveas. Obviously, the fovea is something that happened with the more development of mammalian and primate vision to allow it to be even more sharper than it would be without a fovea. Here's a schematic depicting the distribution of rods and cones across the retina. Now, in the gray images on the left there, you see a photomicrograph of rods and cones actually in the retina. The top one is showing you the distribution of rods. Now, I can tell from the distribution of rods here being so dense that this must be in the periphery of the retina, not in the fovea. The image down below is showing a picture of the retina more toward the fovea, where you've got cones, and the cones are the larger receptors that you see there, those big blobs, next to those small little ones. And the cones are since there's cones here, then this must be close to the fovea. Okay, the graphic on the right is showing the distribution of rods and cones across the retina. It's a real nice graphic to memorize. It's a very easy thing for me to ask test questions about. And it's showing you that the rods are very prevalent in the peripheral parts of the retina, the outside parts of the retina. As you move toward the center of the retina, where the fovea is all cones. So as you move from the peripheral parts toward the center of the retina, toward the fovea, the number of rods is actually increasing as you approach this spot. But there you see that blue line in the middle there, we're showing the cones. In the fovea, there's only cones. And so the number of rods goes down to 0, the number of cones goes to the maximum, and you can see where the cones are really a phenomenon of the fovea. And then the rods are distributed across the retina. But notice that as you go to the periphery of the retina, of course you don't need receptors at all, so even the numbers of rods goes down. So most of the rods and cones are more toward the center, where light's going to be focused. There are only cones in the fovea. And notice also, finally, that of course there's not going to be any receptors where the optic nerve comes out of the retina, because that's an open space where all these neurons are coming out from the ganglion cell layer, and of course, there can't be any receptors there. And so you have a blind spot there. And the book has an excellent little technique for discovering your blind spot. For the life of me, I never knew there was a blind spot until I learned about this. You know, I don't perceive my own blind spot, but you can actually move your head around with a certain kind of stimulus, a certain kind of array, and you'll see things disappear because your blind spot doesn't have any receptors in it. And as you look in that area, you pay attention to that part of your visual field, you'll notice that things come and go. Fortunately, the blind spot, is not anywhere near, I shouldn't say it's not anywhere near the fovea. It's not close to the fovea. And so where your most acute vision is going on, there's no confusion with the blind spot. And your brain actually, I think, kind of fills in what your blind spot leaves out. And so you don't have that sense there's a spot to the right of your vision or to the left for your left eye that's not where something isn't there. Well, the important thing to remember from this graphic is that the fovea has all cones. The number of rods drops off to 0 there. The rods are essentially in the periphery, but that all receptors tend to respond to the fact, tend to be clustered where maximum vision is, even the rods there. They're around the fovea, and as you go toward the periphery, all the receptors drop off. Here's a colored photomicrograph showing the distribution of cones in the fovea. Now these are just the cones, because remember there are no rods in the fovea. And they've colored them by the wavelength that they perceive. So the blue ones are the short wavelengths, the green are the medium wavelengths, and the reds are the longer ones. And the thing you should notice from this is that in the fovea, there are many more cones that perceive the long wavelength light. So they're going to be maximally receptive when the light frequency is in what's referred to as the long or the red range. Here's the distribution of rods and cones in the peripheral parts of the retina, so the outside parts of the retina. The rods are the very small green circles that you see there. Cones are the large green circles. You can barely see the small green circles. You can see them as little dots there, the rods. Cones are the large green circles, and then the large yellow circles are cones that respond to the short wavelengths. These would have been the ones that were coded blue in the previous one. Now notice that in the peripheral parts of the retina, you still have a lot of cones, but you have lots more rods. And so the rods are really a property of the peripheral parts of vision, and cones are a property of the central part. And cones contribute to frequency discrimination, which becomes color, as well as better luminance levels. Here's a slide showing the receptiveness of the different kinds of cones. Remember, there are 3 different kinds of cones, and they tend to be responsive to 3 different frequency ranges. Notice in this graph, though, that they're just maximally responsive. They still respond at other frequencies around these characteristic frequencies, but they're going to respond most when they are hit by light at these base frequencies that they're calibrated to. So the blue cones, and this is blueness, is of course still a property of your brain, not a property of your eyes. The cones that are sensitive to the 430 nanometers are referred to as the blue cones, and you can see them down there. The green cones are responsiveness to the 530 nanometer range, and then redness starts around 560 nanometers. Now what happens is when light hits your retina, a number of these cones will respond. But so imagine a light frequency, say at 500 nanometers hits your eye. Well, the receptors that's going to respond the most will be the green ones, but the others at 500 also respond, especially the red cones. And so what you're going to get sent up into the brain is a pattern of these cones responding depending on what frequencies were of the light that hit it. Your brain then constructs a color out of that, and you perceive greenness or redness or some blending of colors that have a blending of these cones. And so the color you perceive, say it's blue-green, is coming from a light frequency range, I guess, around 500 or so. That would be sort of blue-greenish. Or maybe it's 450. That turns out to be blue-green. Well, your brain gets the information from primarily these two firing, and it interprets that as blue-greenness. And so it maps that part of your visual field in that color, and that's what you perceive. It's a remarkable feat of evolution for this to occur. I mean, it's amazing that your receptors and your brain working together map out a frequency spectrum for you so that you can discriminate objects. Because obviously, if imagine you're looking at a plant, the background color is green, the flowers are red, and you want to be able to pick a flower. Well, the luminance level might be the same for the leaves of the plant as well as the flower. If you can't perceive that the flower is red and the leaves are green, you won't be able to see very clearly where the flower is. It gets all blended together because the luminance level, the intensity level, is the same. You can imagine all sorts of visual arrays in which this occurs, and in which it's hard to discriminate if you can't discriminate colors. And again, I'm going to show you a movie about people who are colorblind, and the trouble they have is exactly this kind of problem. They can't discriminate objects as well as we can, people who have color vision, because they can't perceive these differences across the form of objects. And obviously, discriminating frequency was a great advance in evolution that enabled primates in particular to be very discriminating about the things that they were going to interact with. Things that you're going to eat. If you're, flying through the tree and you're going to grab a branch, you've got to make sure that you can tell where the branch ends and the space begins, because you don't want to grab off into space thinking you're grabbing a branch. Well, at any rate, color perception is a very important part of making these discriminations, and we'll get to that in much more detail in the color section of the course. Here's a graphic that depicts how your lens works. Now, your lens is not made of glass like a camera lens is. It's made out of basically a little bag of fluid. It's very clear fluid, but it is a living thing. It changes with growth and maturation. It's a remarkable invention that you can actually focus light using it. Through the course of evolution, to have something like this develop is just remarkable. Well, what happens is, light enters the lens. It bends the light using refraction, just like a camera lens does. Except the medium, of course, the light's going through is fluid. It's water. And it focuses light onto the fovea. So when you're looking far away, your lens is stretched out. See how it's attenuated there? The little muscles that actually pull it apart and make it thinner. And then light is focused on the retina. Now in the middle is showing a object that's near, in which you can't quite focus on it. It's so close that you can't. The lens can't be accommodated, or it hasn't been yet. And then when it fattens up there, you've got the bottom image. When it fattens up the lens, it bends the light in a way that now focuses on the retina so that you can perceive things using your fovea. Now, most eye problems requiring glasses involve disturbances of the relationship between the lens and how it accommodates like this and the shape of your eye. And I think the next slide goes through some basic problems that most people with glasses have trying to focus light onto their retina. Here's a graphic showing some common eye problems associated with the lens in focusing light on the retina. The top graphic is showing you the normal situation, so that you have a lens that works, your eye is nice and round shaped, and it's associated with the lens, and so when the lens focuses light, it works, everything focuses on the retina. Well, imagine your eyes are not developed correctly, and imagine your eye is a little narrower than it should be. Well, your lens is trying to accommodate to a distance that has developed through the course of evolution, and what happens is it focuses the light past the retina. Now, the person's perception is a very blurry sort of image. Now, sometimes this is relatively mild, and people don't even know that their vision isn't acute; it isn't sharp. Then when they get glasses, they realize, oh my goodness, I can't imagine how I lived before this without seeing so sharply. But look how a convex lens essentially fixes this problem. So now with that convex lens in front of the eye, it will, in a sense, broaden the light so that when it's focused, it's now focused by the lens in the correct way. And people who have glasses know that they go to an optometrist who measures their vision and can tell how far off their lens is, and then make that lens accommodate to the problems that the lens is having with its focus. Now you can have exactly the opposite problem if your eyeball is a little bit too long. So if it's a little bit too long, your lens is going to focus before the retina. And here, a concave lens corrects that problem so that everybody now has their lens focusing light onto the fovea, and their vision now can be completely normal. Now as you get older, your vision changes because your lens begins to deteriorate. And what happens is that cells, as they're in the lens as they die, have difficulty migrating away from the lens, and so they tend to stay there. And over time, the lens becomes stiff. It becomes unable to be, manipulated by the muscles into being squat and narrow. And this is called presbyopia. Presbyopia is a reference to the fact that it occurs with aging. And here you see a graphic showing you the accommodation of the lens over time. And as you can see, people decline pretty quickly over the span of their lifetime. There's a steady decline all the way through the adult years. It tends to stabilize after, it looks like, about age 55 or so. And that's probably when people should get their final set of, bipolar reading glasses. Your eyeball and your lenses can also change in other ways through the course of aging. Sometimes people's vision change, their eyeballs change, their muscles get weak. All kinds of things can happen to make vision and the lens difficult to accommodate. But in general, the process of the lens getting stiffer produces presbyopia, and pretty much everybody is affected by it at some level. Obviously, this is a general trend in the graph. Some people get presbyopia sooner, some later, or they notice it sooner or later, even though it's happening all the time. And then they have to get glasses to correct for it. And usually people get reading glasses, when they're in their 50s. That's usually the time at which they decide, well, I guess I got to get glasses, because you notice them, they'll take a book they're reading and move it further and further from their face, because their lens cannot accommodate to that distance anymore for that fine discrimination that's associated with reading. And so they have to get a lens to put in front of their eye that's going to accommodate for them partially, so the natural lens doesn't have to accommodate so much. Now, one of the things that middle processing layer that involves the horizontal cells is involved in is something referred to as lateral inhibition. And I'll go into lateral inhibition in more detail in different parts of the course that deal with vision. But basically it involves an interaction between the horizontal layer cells that enhance the margins of light and dark. So wherever there's a margin of light and dark, that gets enhanced in the firing pattern of the ganglion cells. So your nervous system gets information that there's a boundary in a certain place. Now this becomes very, very important for mapping contours onto your vision and telling where lines are, because lines make up forms. And what we mostly want to do with vision is interact with objects in the world that have form. And so when we process visual information, we want to enhance the margin of light and dark, because that's a clue that there's a form there. There's an object there, a margin. It doesn't make any sense to know an apple's in front of you if you don't know exactly where its margins are, because you won't know where to grab it. You won't know where to find it. You won't know where to manipulate it with your hands if you can't see where the margins and lines are. So the perception of lines and contours is extremely important in perception, and the lateral inhibition that happens in the retina, because it's going to sharpen the image, essentially like a Photoshop, sharpening filter. It sharpens the image so that you can see margins better. And it partially does that through lateral inhibition, but also a lot of processing in the central nervous system. But the first phase of that, since it happens in the retina, is part of this initial introduction to the retina and your eye.